It's a great pleasure and honor to introduce Kornelia Nelly Polyak as the recipient of one of this year's Paul Marx Prizes. Nelly obtained her MD degree in May of 1991 at the Albert Szent-Györgyi University in Szeged, in her native Hungary. In May of 1991, she joined then in September of that year the graduate program at the Cornell Medical School, Graduate School of Medical Sciences, before it was the Weill Cornell Medical School of Medical Sciences. She joined my lab. I was very fortunate with that incorporation into the lab, because it was the courage of Nelly, which is one of her distinguishing features as an investigator, her courage to plunge into what then was or looked like a daunting task, which was to purify biochemically P27, what turned out to be P27, a cyclin-dependent kinase inhibitor discovered for its ability to somehow mediate the tumor suppressive and growth suppressive actions of TGF-beta and cell-cell contact inhibition. Not only did she clone it without any formal training in biochemistry, in protein purification, but she went on to clone it. And in barely 4 years, she graduated from the PhD program and moved on, now inspired by the relevance of tumor suppressor genes and their biochemical mediators, moved to the lab of Verv Augustin and his colleagues at Johns Hopkins, where she conducted a postdoctoral training, now approaching tumor suppression with a more genetic flair to it. And that was at the time when others, also being honored today, had developed SAGE, the system for analysis of gene expression that the lab developed. And she was one of the first to apply SAGE to analyzing a process of cell suppression by P53. That was, again, a spectacular career, highly productive. And with that, she arrived as a faculty member to the Dana-Farber Cancer Institute, where she has remained ever since. Again, driven by her courage, fearless attitude towards plunging into the grit of complex biology, especially as it relates to cancer. She took on to analyze, profile, and try to explain the nature and significance of the heterogeneity of tumors, especially breast tumors, breast cancers, analyzing both the cancer cell population and the stroma cell population, and comparing it to the heterogeneity of these cells in normal tissues. This is very important work that very few would have the guts and courage to take on. But Nelly did. And so for that and her previous accomplishments, we are very pleased to honor you today, and welcome to this podium.
So thank you. I'm just recovering from a cold, so hopefully my voice will survive. But I'm very honored to be a recipient of the Paul Marx Prize, and for me it has a special meaning, having started my career here, as Joan mentioned, in his lab. And that very, very exciting time, which I think very few graduate students are fortunate to experience, really committed me to stay in research and continue my career and research training. So I'm, first of all, I have to thank Joan for that. And also that was a time that Paul Marx was still the president, and we actually collaborated with his lab. So it was nice to see the work also, to go from basic science to clinical translation, and that kind of guided me to stay in that direction. I also have a few other people to thank. My family, who stays outside because a toddler is not appropriate for a lecture room, but they have been really, really great for helping me through the years and always supporting whatever I decided to do, whether moving across the continents and so on. So it has been very, very good for me. Then Joan, my PhD mentor, who took the risk of taking somebody arriving straight from Hungary and embarking in a really, really important and competitive project, which worked out pretty well, fortunately, but that was a really, really exciting time to me. Then my postdoctoral mentor, Bert Vogelstein, who's another outstanding scientist who really told us, all of us in the lab, that you have to focus on a patient. That's the ultimate goal of cancer research, is to cure somebody of their disease. As many of you who know him knows that he's a very humble person. He always said that, until you don't reach that point, prizes publications are not really showing you that you achieved your goal. So it was a great experience to work with him. Then David Nathan, who's actually flown down from Dana-Farber, is another person I have to be thankful, because he was the president of Dana-Farber at that time. And he was very, again, courageous to hire me as one of the youngest faculty, as I learned from at that time. And I still remember David asked me when I was interviewing that, "Are you really ready for this?" And I said, of course, yes. But as many of you know, you're going through the training and you always feel like you're ready for the next step, but you really know whether you're ready, what it is when you're there. But again, fortunately, I think it worked out pretty well, and we learned a lot during the years. And then another mentor I have here in the room, Mina Bissell, who's sitting in the back, who I had the fortune to interact with over the past several years. And many of you know Mina. She's really one of the most sparkling and energetic scientists. And particularly, she's been emphasizing and helping us to putting cancer into context in a way that looking at the tumor as a whole, and not just individual cells, tumor cells alone. And I've been very grateful to have her as a mentor and also as a friend over the years. So with that introduction, I like to tell you some of the highlights of the work we did over the years, without really going into much detail, because we got the instruction of having a 20-minute talk, and I was trying to follow that. But basically, when I started my work, and over the past over 10 years now, we wanted to understand what makes breast tumors develop, what are the changes that occur from a normal breast tissue to DCIS, which is ductal carcinoma in situ, that progress to invasive tumors and metastatic tumors. And this slide also summarizes our approach and kind of understanding the complexity of this disease progression, because initially we started out looking at the tumor epithelial cells, which, classically, the mutations accumulating in these cells were thought to be the drivers of tumorigenesis. And I will show you some data that basically we haven't been able to find any genetic changes that distinguishes DCIS and invasive tumors that could explain this progression. And that was the finding of many other labs as well. So then we actually realized that tumor cells don't sit on their own in the tissues, but they actually form these complicated cell-cell interactions. And if we look at the interactions that occur in a normal mammary epithelium, where you have a ductal epithelial cells that are red, surrounded by the myoepithelial cells that are blue, and then they form this basement membrane, one of the most dramatic changes that occur as the tumors progress is that these changes in the microenvironment. And particularly from a transition from in situ to invasive carcinoma, the disappearance of the myoepithelial cell layer, and the cell layer actually forms the basement membrane, is what's the distinguishing feature. And it's one of the most critical progression points, because DCIS is still within the duct, and it's curable largely just by surgery. Whereas as the tumor progresses, then you're already facing a much more complicated challenge of curing the spread tumor cells. Then as we're studying the tumors, we actually nowadays reached the conclusion that tumors don't really follow these very strict progression steps that we would like to see. Particularly if you're looking at the tumor epithelium, it doesn't look like that you have always this clonal succession of cells acquiring more and more changes and always the same population dominating the tumor. But in reality, it's a much more complicated picture in a way that you can have multiple clones coexisting in a tumor, and these tumor cells, not always the same clone that's dominant in one stage will be the dominant in later stages. And more importantly, the treatment can also change and favor the outgrowth of one or the other clones as well. So this kind of leads to a much more complicated picture of trying to understand tumor progression. So very briefly, I will show you some highlights from each of these areas that we learned over the years. So I mentioned that we initially started our studies with looking at tumor epithelium cells, and we purified from different stages, normal, in-situ, invasive carcinoma, and did gene expression profiling, also looking at genetic changes by CGH, SNP arrays, sequencing candidate genes, and basically we found no distinguishing feature that would classify DCIS and invasive tumors into different stages. The same for genetic alterations. If you compare DCIS to invasive tumors, essentially you see the same changes. The degree of them, frequency may be different, but there is not really like a particular gain or loss that would differentiate the tumors. So then I mentioned that the tumors and the normal tissue is composed of many different cell types, not just tumor epithelial cells. So then we decided to purify each one of these cells, as many as we could, from fresh tissue samples. Each color is a different cell type, and we purify these cells to as homogeneous populations as we could, using these magnetic beads coupled to cell surface markers specific for the particular cells. And then again, we did profiling of these distinct cell populations. What we learned initially, like on the gene expression profiles, is that actually not just the tumor epithelium, which is in red, is what's changing during progression, but also the various other non-malignant cells, such as the myoepithelial cells, the myofibroblasts, even the regular fibroblast and leukocyte populations seem to be very different if you compare normal DCIS and invasive breast tumors. Genetic changes, which are one of the hallmarks of cancer, we only found in tumor epithelial cells. This was by SNP arrays looking at LOH, loss of heterozygosity. Blue areas mark those regions that lost, and as you see again, the coloring shows that these clonally selected genetic changes seem to occur only in the tumor epithelium, and we actually had one kind of a strange breast tumor, phylloides tumor, which has both malignant stroma and malignant epithelium. In this case, both were malignant, so showing that the technique of purification of the cells was not biasing against detecting these changes. But then we also knew that the cells that compose the microenvironment, they permanently seem to be altered. So if not genetic changes, then we thought that maybe there's epigenetic alteration that may be responsible for maintaining these abnormal properties of the tumor cells. So we actually, this was like 6 years ago, we wanted to investigate global DNA methylation patterns in these various cell types during tumor progression. So we developed this technique, which we call methylation-specific digital karyotyping. At that time, we could not just sequence the whole genome, which is basically a sequencing-based approach, very similar to SAGE, except that we use genomic DNA, cut it with a methylation-sensitive restriction enzyme, derive fragments, sequence those fragments, and then based on the counting of those fragments, we can tell whether methylation is occurring or not. So we applied this technology to look at the non-epithelial cells and epithelial cells in tumor progression stages. And this graph just shows the statistically differentially methylated loci. Each dot is a particular gene, and these are chromosome 1 through X. And we see many changes in a tumor epithelium if we compare normal to tumor, but also in the myoepithelial cells, and I don't show it on slide, but even in a fibroblast. In the tumor-associated fibroblasts seem to be epigenetically different from the normal fibroblast. So then based on these studies, we concluded that we see all these gene expression and epigenetic changes in the microenvironment. Genetic changes also only occur in a tumor epithelial cells. But the question is this only a reaction to the tumor, meaning like these microenvironmental changes, not just a reflection of tumor involvement and not really playing a really relevant functional role? So it's not easy to address that in humans, because humans, you can only do association studies. So we used a model of DCIS to address this question whether the DCIS to invasive progression is really like an escape mode, meaning like the tumor epithelial cells gain something that makes them able to break out this barrier, or maybe it's more complex and actually there is a barrier breakdown, which kind of more like a release mode of progression. And we decided to use a xenograft model of DCIS, which has limitations, but this particular model, the MCF10 DCIS, seemed to reproduce the DCIS, human DCIS, fairly well, because we see these very nice DCIS-looking lesions. Smooth mesolectin and P63 forms this myoepithelial layer. And then these tumors progress to invasive tumors within a few weeks. So basically, it reproduces the histology and also the natural history of DCIS. So we use this model to address the role of the microenvironment, particularly the myoepithelial cells and the fibroblasts in this progression. What we did initially is do co-injection studies, mix these epithelial cells with human myoepithelial cells, which is HME, and we found that they very potently suppress tumor growth. When we mix them with fibroblasts derived from normal breast or rheumatoid arthritis or inflammatory fibroblast from actually breast tumor and an inflammatory fibroblast from rheumatoid arthritis, what we found that these very potently promoted tumor growth. The rheumatoid arthritis we actually decided to use because when we did our initial profiling studies with breast tumor-associated fibroblasts and looked at some other literature on fibroblasts, we found that the rheumatoid arthritis-associated fibroblasts very much look like tumor-associated fibroblasts, again highlighting this notion that tumors and inflammation has a lot in common. In addition to the tumor rate, we also noticed that the fibroblasts seem to promote invasive progression because even at these early stages when the DCIS was still DCIS-looking tumors, whenever we co-injected any of the fibroblasts, this myoepithelial cell layer completely disappeared, and these tumors were already invasive at this early stage. The myoepithelial cells were able to overcome the effect of fibroblasts because when we did triple injection, every single time the tumors were suppressed, again showing that this myoepithelial cell layer is really important for preventing tumor progression. And additionally, the histology of the tumors was also, again, retained more DCIS when we co-injected the normal myoepithelial cells, really highlighting the importance of this myoepithelial cell type in the preventing progression to invasive tumors. In terms of looking at what are the changes that occur in the tumor epithelium in response to the stroma, we found that the COX-2 enzyme seems to be very highly overexpressed only when we co-inject the cells with the fibroblast. And this actually could induce angiogenic genes, potentially explaining why those tumors grow faster. But this also gave us the opportunity to test, can we abrogate the tumor-promoting effect of the fibroblast by treating with a COX-2 inhibitor, which we did in a mice. So basically, we had the control animals co-injected with the rheumatoid arthritis fibroblasts. We see an increase in tumor rate and also increased progression to invasion. When we fed the mice with a COX-2 inhibitor, then we could completely eliminate this tumor-promoting effect of the fibroblast, and also the histology was more resembling DCIS histology, suggesting that these COX-2 inhibitors may be used as a prevention, not just the prevention of tumor initiation, but also the progression to invasion. And just to summarize, basically, we have many ongoing studies further dissecting the role of the myoepithelial cells in this progression. Basically, our hypothesis now, we have some data that support that in DCIS, the differentiation of the progenitors to myoepithelial cells is somehow compromised by signals coming from a microenvironment and also the tumor epithelial cells, and we progressively lose these differentiated myoepithelial cells, which are the ones that produce the basement membrane. So basically, by losing this cell type, the physical barrier breaks down and the tumor becomes an invasive tumor. So that highlights that this interaction among the cell types and not just the genetic changes in the tumor epithelial cells that eventually influence progression. Another area that we became interested in a couple of years ago is this heterogeneity of the tumor cells, the tumor epithelium itself. And a few years ago, this was now actually 6, 7 years ago, there was this cancer stem cell model proposed, and in a breast, CD24 and CD44 were proposed to be markers of the more differentiated and more stem cell looking cells. So we decided to purify these cells from multiple tumors and normal breast tissue as well. This shows the gene expression profiles that very differentiates the CD24, CD44 cells, and really looks like that the CD44 cells express a lot of markers common for stem cells, and also markers of angiogenesis, invasion, and so on, suggesting that they really look more stem cell-like and more likely to play a role in invasive and metastatic progression, whereas the CD24 cells were more luminal epithelial cells. But as you know, like, cancer is a genetic disease, so we also wanted to question is, like, we have these two different populations, but are they genetically homogeneous or not? And well, we found actually surprisingly they are not. So there is in addition to having this phenotypic heterogeneity of the stem cell and more differentiated phenotypes, there is a genetic heterogeneity as well, as this is just an example of looking at some amplifications of AFQ24. The CD24 cells, CD44 cells both have multiple copies. But when we look at another change, 1Q21, these 44 cells only have 2 copies, whereas the 24s have multiple copies. So that suggests that there is not a really simple stem cell differentiated cell relationship, because in addition, there are these genetic diversity within the tumors. And this area we explored in more detail in the past years, looking at the genetic and phenotypic heterogeneity in the tumors as they progress. This particular study, we looked at DCIS, comparing DCIS into invasive tumors, and using this technology, immunofish, which basically you stain for markers, CD24, 44 here, what we used, and also do FISH for commonly gained regions. And then just by looking at the pictures, you can tell that there is a very high degree of heterogeneity, both for the marker expression and also for copy number gain for these particular loci. And for example, in some cases, you can really see that there is a subclone of cancer cells that seem to take over tumor as they progress. For example, in this particular tumor, we saw a gain of 11Q in a subset of cancer cells in a DCIS in a 44 plus fraction. But then in an invasive tumor, both the CD24 and 44 plus cells seem to have this gain, suggesting that this clone took over, and also shows that these phenotypes can change in the tumor. We wanted to derive a score that reflects the diversity of the tumor, not just by looking at the cells, but actually the one number that could be used kind of as a biomarker for tumor diversity. And this index, which called the Shannon index, is a terminology that ecologists really use to assess biodiversity in nature. So when we calculated this index in different types of breast tumors, one would be like a normal level that you can see even in normal tissues. What we see that the basal-like breast tumors, which is actually one of the worst type of breast cancer in terms of very difficult to treat, they always have high diversity, whereas the HER2 plus and luminals, sometimes they have low, sometimes they have high diversity. And we're currently doing studies looking at if this diversity is actually a predictor of clinical outcome. And I know there is some data in sequencing studies that it looks like that clonal diversity most likely predicts therapeutic failure. We have been looking at these diversity changes in metastatic lesions, and it's actually very kind of discouraging in a way that if we compare 2 different metastatic lesions in the same patients, again, like we see a very high degree of divergence, both genetic, this particular lesion has a gain of this chromosomal region, this does not, and then also the phenotype seems to be very different. Similarly, we are addressing whether the treatment itself changes heterogeneity and the phenotype of the tumor cells by comparing pre- and post-treatment samples. And this is still ongoing, but it looks like, again, like that the treatment itself has a dramatic effect how the cells populations behave. So then coming back to this heterogeneity, it seems very discouraging. You know, if the tumors are this heterogeneous, how can we treat them effectively? Particularly, are these CD24 and CD44 cells have some dependencies that will differentiate them. And sorry, my voice is going away, so I will finish soon. We addressed this by conducting an shRNA screen for taking the genes that were differentiating these two cell types and asking which one would be required for any particular cell type. And we identified 15 genes that seems to be particularly important in these stem cell-looking basal-like CD44 plus cells. I don't cut it short because I don't have much time, but basically what we found that one of the pathways, this IL-6 JAK2 STAT3 pathway, seems to be particularly important in basal-like breast cancer cells which have this CD44 phenotype. This shows a xenograft assay showing that with a JAK2 inhibitor, we can very potently suppress the tumor growth in mice. And also even in tumors where we don't see very dramatic shrinkage, the cellularity of the tumor is very potently suppressed. In these breast tumors, we actually looked at that if we assess the frequency of the STAT3 positive cells within tumors in terms of like marker CD44 and CD24 expression, it looks like that the CD44 plus CD24 minus cells are the most frequently phospho-STAT3 positive, whereas the CD24 negative, positive 44 negative cells are very rarely positive, and these are just a quantification of this. This is based on 170 invasive breast tumors. And this cell type, which is this 44 plus STAT3 positive cell, again, it's present in all subtypes, but it's the most frequent in a basal-like breast cancer. And actually, based on these studies, we're initiating a clinical trial now testing JAK2 inhibitors in these tumors that have phospho-STAT3 positivity. So just to conclude, basically what I was trying to show you, that we think that this tumor progression is a very much like an evolutionary process that's driven by genetic, epigenetic, and phenotypic diversity in the tumors, combined with a selection by the microenvironment. And the microenvironmental changes also seem to be playing an important role in this progression. And we think that better understanding this diversity of the tumors, both in terms of tumor cell diversity and the microenvironmental diversity, that will lead to more efficacious treatment. An area which I haven't had time to tell you about, we're also looking at normal breast tissue, and particularly trying to understand why women who develop breast cancer may already have changes even before they have cancer in the normal tissue that could be used to maybe risk stratification, and also identifying those with high risk and particularly targeting with preventative strategies. So I just like to thank the most important people who helped me get this prize, which are the fellows and the trainees in the lab. These are the past lab members and the current lab members that have been really, really instrumental to generate all the data and experiment that I showed you today. And also we have a large number of collaborators throughout the world, different countries that basically have been helping us over the years. And I can't read all the names, but we have been working very extensively with literally people from different continents. And nowadays with Skype, you can basically collaborate with anybody, anywhere, and we can have virtual conferences in one room despite being in different, the time zone is still a difference, the problem, but otherwise it's technically impossible. Then I also already mentioned where Joanne and the lab where I started my career, particularly Francis, who was also a graduate student, Fernando, and Jeff Rana, who helped me, is actually a prior recipient of the prize as well, but they've been really, really instrumental to help me develop as a, scientist, I still remember Jeff told me, like, you can never have enough controls in any experiment. And that's actually very true. And I'm glad that he was so helpful during my graduate studies. Then my postdoctoral mentors, Bert Vogelstein and Ken Kitzler, where I did my training. This was the blizzard of '95, I think. And then Victor, this was the SAGE group. And they had to sit down because all of them are like, as you see Victor, like 6 feet or something, and I'm like a dwarf next to it. So I refuse to have a picture where we're standing. That's why we have it all sitting there. And it's been a great, great experience. And again, like, very thankful to them. And that's it. And fortunately, my voice still remains somewhat.
In light of the hour and the state of Nellie's voice, we will take 1 or 2 questions, especially of the kind that can be answered with a yes or a no. How much is stochastic event at play in this, even in tumors of a certain kind? Mm-hmm. You have a great deal of diversity, heterogeneity in anything you measure. So how much is, you think, chance? Very high, very low? You can answer high or low.
So I think it's in terms of, acquiring mutations and all that is a stochastic event. So I think a tumor is not really like thinking about what happens. You have cells that are different, and some are different in genetic terms, and then you always have a selection. So, and even the progression, I think there is a lot of stochastic in terms of, like, some tumor cells can get out of the tumor, but it doesn't mean that that will be the cell that actually develops metastatic lesion or not at a time. So I think there is a lot, yeah.